Imagine those moments when a word teeters on the tip of your tongue, or maybe a familiar face feels just out of reach for a second. You might wonder, is this just, a normal part of getting older, or is something more significant happening? For millions, sadly, these subtle shifts are the first quiet whispers of a relentless thief. Alzheimer's disease. It's a disease that doesn't just steal memories. It really erodes independence, connection, and ultimately identity. For decades, the earliest molecular changes in Alzheimer's have been kind of shrouded in mystery, a really complex biological puzzle. Scientists have been desperately trying to solve it. The frustrating truth is we haven't had a truly proactive, accessible way to intervene at those critical early stages. But what if a tiny, often overlooked element, something naturally present in our bodies, in our environment, holds a significant, maybe even treatable key? A key to preserving our cognitive health and potentially preventing the onset of this devastating disease. That feels truly revolutionary. Okay, so let's unpack this. I'm genuinely curious. Today, we're diving into truly groundbreaking research. It's from a team of Harvard researchers, just published in the prestigious journal Nature. And this isn't just, another scientific paper. It feels like a potential game changer. It points to a surprising yet possibly treatable root cause, something that impacts cognitive preservation during aging, and could be crucial in halting the very initiation of Alzheimer's disease itself.
Exactly. And what's truly fascinating here, I think, is that this research offers so much more than just identifying new risk factors, which is often what we see. It really delves into a fundamental understanding of the pathogenic processes, the how and why the disease actually starts. And importantly, it presents genuinely practical, kind of easy to understand insights, things that can make a profound difference in how we approach brain health and aging. It feels like it's about finding real solutions, not just more questions.
Right. So our mission today is to try and cut through any scientific jargon. Let's extract the most important nuggets of knowledge from this complex study and present them in a way that leaves you feeling truly well-informed, maybe even a little surprised, and most importantly, empowered about this critical new development in brain health. We really want you to walk away understanding not just what happened in the lab, but what it could actually mean for your future, for our future. So the scientific journey, it didn't start with lithium specifically in mind, right? It began more like a detective investigation into the brain's internal chemistry. Researchers meticulously examined the balance of various metals, not just the toxic ones we hear about, but also those vital for healthy brain function. And what they uncovered was, well, you said it, singular. It pointed them directly to one surprising suspect.
That's absolutely right. Imagine being able to scan a brain for every single tiny mineral with incredible precision. That's essentially what they managed to do. They used these advanced techniques, specifically something called inductively coupled plasma mass spectrometry. It's a mouthful, ICP-MS for short. Think of it like a super sensitive fingerprint reader, but for chemical elements. This allowed them to precisely analyze 27 different abundant and trace metals, and they looked at samples from post-mortem brain and blood, very carefully chosen from individuals across the entire cognitive spectrum. So people with no cognitive impairment, NCI. Those with mild cognitive impairment, MCI.
Which is often that very early precursor stage.
Exactly, that crucial early precursor stage. And of course, individuals with full established Alzheimer's disease, AD. Now, the specifics of their findings were what made this discovery so striking. So, as you said, singular. Out of all those 27 metals they surveyed, only one showed significantly reduced levels, and it was in a very specific area, the prefrontal cortex.
The decision-making center.
Right. Critical for executive functions, decision-making, memory, personality, and it's known to be prominently affected very early on in Alzheimer's disease. This reduction wasn't just in established AD, but crucially also in those with MCI, that early vulnerable stage.
So if I'm understanding this correctly, it wasn't like a general depletion across the whole brain. It was highly targeted, right in the region where we see the earliest signs of Alzheimer's starting.
Exactly. And that distinction is incredibly important. It wasn't random. This reduction was specifically localized to the prefrontal cortex, that vulnerable region, but not in the cerebellum, which is a brain region relatively unaffected in Alzheimer's.
Okay.
So that geographical specificity within the brain immediately told them something profound was happening. And further supporting this, they looked at lithium levels in the blood serum lithium, and they found that the mean serum lithium levels in MCI and AD patients were not significantly different from healthy controls.
So it's not a whole body issue then.
Precisely. It underscores that what we're talking about is a brain-specific deficiency. It's not a systemic blood issue that you could just pick up on a standard blood test. Think about it this way, maybe. Your car starts sputtering. A quick check of the oil and the entire system shows it's fine, levels are good. But if a mechanic specifically looks inside the engine's most critical carburetor, they might find a tiny essential component slowly disappearing.
Causing the whole thing to malfunction.
Exactly, causing the entire engine to malfunction. That's the parallel here. A distinct reduction of an essential element, lithium, specifically in the brain regions most vulnerable to Alzheimer's, even at its earliest stages. This insight right from the start was a huge aha moment for the researchers.
Okay, so once they knew lithium was specifically disappearing from these critical brain regions, the natural and pretty urgent question is why? Why does this vital lithium vanish from the brain or become so deficient right where it's needed most? What did the researchers think was causing this?
That's precisely the question they asked next. And what they found was, well, truly insidious. It turns out the very plaques that are the pathological hallmarks of Alzheimer, like, those sticky amyloid beta deposits, ABO shoes, we've heard so much about them, they might actually be holding the lithium hostage.
Hostage? Really?
Yeah. The research revealed a striking and highly significant concentration of lithium within these amyloid plaques themselves. This wasn't just a theory. They observed it both in human MCI and AD cases. And importantly, they replicated it in AD mouse models. What's more, this concentration of lithium within the plaques actually increased as the disease progressed, from MCI to full AD.
Wow. So the disease isn't just happening around the plaques. The plaques themselves are actively contributing to the problem by trapping this essential element. That's a different angle than I usually hear discussed.
It's a critical new understanding, I think, to get into the scientific detail just a bit. They used an even more sophisticated version of their technique, laser absorption ICP-MS. This allowed them to literally pinpoint and measure elements within specific tiny regions of the brain tissue, like microscopic mapping.
Amazing.
And with this, they showed about a 3- to 4-fold concentration of lithium directly within the plaques compared to the plaque-free regions nearby in the mouse models. This process, they termed it sequestration. These amyloid deposits are essentially acting like tiny biological black holes soaking up and trapping lithium. This effectively reduces its bioavailability, its ability to be used in the active non-plaque parts of the brain. These are the parts where lithium is actually needed for healthy function, for neurons to communicate and thrive.
So it's like the brain needs this vital nutrient,
Yeah,
but these sticky plaques are acting like sponges, just soaking it up and trapping it.
exactly. Aggressively soaking it up, preventing it from reaching the cells that need it most. It's creating a localized famine right in the heart of the most important cognitive areas. And this isn't just an observation, it has direct functional consequences. This is really important. The study found that lower lithium levels in these non-plaque cortical fractions, the available lithium, correlated directly with reduced cognitive test scores.
In people.
In people, yes. Across the Asian population they studied. This included scores for episodic memory, like remembering recent events, semantic memory, your knowledge of facts, and a global index of cognitive function. And in the AD patients specifically, this correlation was particularly strong for episodic memory and global cognitive function.
So less available lithium meant poorer memory and thinking abilities. It's a direct link.
A direct measurable link.
That's a powerful and frankly alarming connection. Okay, so to move beyond just correlation, to really understand cause and effect, to prove that lithium deficiency wasn't just associated with AD, but actively driving it, what was the next logical step for the Harvard team? How did they test that?
Right. This was the crucial step to prove causation, not just correlation. The Harvard team designed these meticulous experiments. They looked at what happens when lithium is intentionally reduced. They did this in the diet of both wild-type mice, so normal, healthy mice, without any genetic tendency towards AD.
Kind of like a control group.
Exactly. And also in established AD mouse models, which are specifically engineered to develop Alzheimer's-like pathology. By selectively removing lithium from the mouse diet, quite a significant reduction, 92% they managed to achieve about a 50% reduction in cortical lithium levels in the brain. This precisely mirrored the deficiency they had observed in the human brains they studied earlier.
Okay.
And this drastic, yet very controlled reduction in dietary lithium had profound, widespread effects. In the AD mouse models, like the ones called 3XTG and J20 lines, these are standard models used in labs to study Alzheimer's. Reducing their natural cortical lithium levels markedly increased the deposition of amyloid beta, and it also increased the accumulation of phosphatau,
The plaques and tangles.
the two classic toxic protein aggregates that define Alzheimer's disease. Exactly. And this increased plaque burden and tangle formation happened remarkably early. We're talking within just 5 weeks of the mice being on this lithium-deficient diet.
5 weeks? That's incredibly fast.
Astonishingly rapid. It really shows just how critical lithium seems to be for keeping these processes in check.
So it's not just the subtle effect then. A lack of lithium actively speeds up the development of the disease's hallmarks. But I imagine it wasn't just the protein aggregates, was it? A problem this fundamental usually has broader ramifications.
You're absolutely right. The impact was much broader and frankly more devastating than just the proteins. Beyond the classic plaques and tangles, lithium deficiency also led to a significant activation of microglia.
The brain's immune cells.
Exactly, but not in a good way. They shifted into a pro-inflammatory state. They started causing damage instead of protecting the brain. There was also a substantial loss of synapses.
The connections between brain cells.
Yes, the crucial connection points, the hubs of communication, as well as axons, the wires of the brain. And myelin, that protective fatty sheath around the wires that allows for efficient, rapid signal transmission.
Wow, so the whole infrastructure.
A complete breakdown of the brain's communication infrastructure. All of these degradations are absolutely central to AD pathology and cognitive decline. And crucially, these widespread pathological changes translated directly into accelerated cognitive decline. This included significant memory loss. And this wasn't just in the AD mouse models. Remarkably, even the aging wild-type mice, the ones that normally don't develop AD-type pathology, showed memory loss on this diet.
Just from lack of lithium.
Just from the lack of lithium. This controlled experiment is incredibly powerful. It demonstrates that lithium isn't just passively low in AD. Its deficiency actively drives the disease's progression. It affects multiple critical aspects of brain health, from the buildup of toxic proteins right down to the very ability to think, learn, and remember. It's like a cascading failure, a domino effect, all directly linked to the absence of this single tiny element.
So what we're learning is that if lithium is deficient in the brain, it's not just a minor nutrient imbalance. It's actively accelerating the very hallmarks of Alzheimer's. It's creating this catastrophic internal environment. Let's really dig into what that cascade of decline looks like. What are the specific consequences?
Precisely. The study meticulously detailed how this lithium deficiency directly fuels the accumulation of both amyloid beta, APIA, and phosphatase, those toxic proteins forming plaques and tangles, respectively. In one specific model, the 3xTG80 mouse model, which develops both types of pathology, they saw a massive increase. Both early-stage phosphatase and the more advanced-stage phosphatase were increased 3 to 4-fold. And this was specifically in the hippocampal neurons.
A memory center.
Exactly. That critical seahorse-shaped structure deep in your brain, fundamental for forming new memories. And it's one of the very first targets in Alzheimer's disease. And again, this wasn't just a problem for mice genetically predisposed to AD, that's key. They found an elevation in 842 that's the particularly sticky and toxic version of amyloid beta, considered the major pathogenic species in AD. Even in aging wild-type mice on a lithium-deficient diet. This tells us something profound. Lithium's protective role seems to extend beyond just established AD models. It critically impacts normal aging processes as well. It's not merely a bystander effect. Lithium appears to be an active player in regulating the formation of these toxic proteins, preventing them from building up. Its absence doesn't just allow them to proliferate, it seems to actively encourage it.
And when we talk about Alzheimer's, what I think many people fear most is that erosion of memory, their sense of self. So what about the impact of this lithium deficiency on those most cherished cognitive abilities? What did the researchers actually see in terms of memory loss?
Well, the behavioral assessments were stark. They provided some of the most compelling evidence, I think. The administration of the lithium-deficient diet significantly impaired learning and long-term memory in the AD mouse models. And this wasn't just a vague observation. It was rigorously measured using standard, widely accepted tests in neuroscience. For example, the Morris Water Maze. That assesses spatial learning and memory. Think of it like remembering how to navigate a familiar neighborhood.
Right.
The Y Maze looks at spatial working memory, like remembering where you just put your keys down. And novel object recognition tests, which evaluate recognition memory, like recognizing a new face or an object you've seen just once before. Across the board, the mice on the lithium-deficient diet performed dramatically worse in all these tests. And what's even more noteworthy, and really highlights the broader implications here, is that the aging wild-type mice, remember, these are the mice without a genetic predisposition to AD. They also showed significant memory loss in these same cognitive tests when they were on the lithium-deficient diet. Importantly, these cognitive deficits occurred without any changes in their swimming speed or general exploratory behavior. That rules out the possibility that the mice were simply less active or maybe had general motor problems.
So specific to thinking and memory.
Exactly. It indicates a direct, specific effect on cognitive pathways, on the very ability to form and retrieve memories.
So could that potentially mean that adequate lithium levels aren't just protecting against Alzheimer's specifically, but are maybe crucial for maintaining sharp cognitive function and memory as we age, just generally?
Absolutely. That's a major implication here. It means endogenous lithium, the lithium naturally in your brain, protects against memory loss, not only in the context of AD-type pathology, but also during what we consider normal aging. It strongly suggests a direct link between adequate natural lithium levels and maintaining sharp cognitive function, overall cognitive resilience as we get older, regardless of whether someone has a specific AD diagnosis or not. It's about the fundamental health of our brain's operating system.
And speaking of the operating system, this deficiency isn't just hitting one target, is it? It seems like it's affecting the very deepest, most fundamental blueprint of the brain cells themselves, almost like disrupting their genetic instructions.
Indeed. And to understand this deep impact, the researchers use a really cutting-edge technique called single-nucleus RNA sequencing, or snRNA-seq. This allowed them to look at gene expression changes, essentially, which genes are turned on or off at the level of individual cells within the hippocampus, that critical memory center. And what they found was truly alarming. They saw significant transcriptome changes, meaning widespread alterations in gene expression across multiple brain cell types. And this happened within just 5 weeks of lithium deficiency. That's a very rapid and broad impact on the brain's most basic functions. These affected cell types included excitatory neurons, granule cells, inhibitory neurons, the main communicating cells of the brain, but also oligodendrocytes, which produce the protective myelin sheath, astrocytes, which are crucial supportive cells, and microglia, the brain's immune cells. It was across the board.
Pretty much everything.
Pretty much everything. For instance, in excitatory neurons, genes related to synaptic signaling and organization, essentially, the genes that tell neurons how to communicate, connect, build their circuits were downregulated. They were turned down, muted. And at the exact same time, pathways associated with neurodegeneration and Alzheimer's disease were upregulated, actively turned on.
A devastating double hit.
Exactly. The brain's normal, healthy functions are suppressed, while disease-promoting pathways are activated.
It's like you said before, like lithium is a master conductor for the brain's cellular orchestra. And when it's missing, entire sections of the orchestra just play out of tune, disrupting the whole harmony, the whole function.
It's precisely that, a really good analogy. And critically, the transcriptome changes they observed in these lithium-deficient mice broadly overlapped with gene expression changes found in human cortical biopsy samples from people with AD pathology.
Ah, the human connection.
Exactly. This concordance provides powerful evidence that the mouse model findings are highly relevant to human disease. It suggests that the same underlying cellular processes, the same out-of-tune sections of the orchestra, are likely at play in human Alzheimer's.
So if this cellular blueprint is disrupted, what does that mean for the actual physical structure of the brain, for its ability to connect and communicate? Are we talking about real physical decay here?
We are, quite literally. It leads to significant structural degradation. The study found that lithium deficiency broadly downregulated genes involved in synaptic signaling and structure, meaning the genetic instructions for building and maintaining these crucial connections were essentially turned down or off. This disruption manifested physically as detectable dendritic spine loss.
Those little receiving antennae on the neurons.
Exactly. The tiny mushroom-shaped protrusions on neurons that receive signals, they were disappearing. And they saw reduced levels of essential presynaptic and postsynaptic proteins, like synaptophysin and PSD95. Think of these proteins as the specialized glue and machinery vital for the proper function and stability of synapses. So their reduction indicates a real breakdown in those critical communication points. And beyond the synapses, the study also showed a significant loss of myelin.
The insulation around the wires.
Right, that fatty sheath insulating axons, allowing for rapid electrical signal transmission. Imagine the plastic coating around an electrical wire. Without it, the signal is slow, inefficient, prone to short-circuiting. This loss was accompanied by reduced numbers of oligodendrocytes, the specialized cells that actually produce the myelin, and even a direct reduction in the number of axons themselves, the wires.
So the brain's critical wiring, its infrastructure, is literally decaying and becoming less efficient. That sounds like a recipe for slowed thinking, lost connections, memory problems.
Exactly. They confirmed this with transmission electron microscopy, which gives us ultrafine microscopic detail. It showed thinner myelin sheaths and something called an elevated G ratio, which is a technical measure consistent with reduced axonal myelination. It's just like you said. The critical wiring in a complex electronic system becoming exposed, thinned, inefficient, leading to widespread communication breakdowns. So endogenous lithium contributes directly to maintaining the structural integrity of synapses and myelin, both absolutely crucial for learning, memory, and efficient brain function. Its absence means the brain's physical wiring begins to fray.
Now, the brain also has its own dedicated immune system, right? The microglia. They're supposed to protect it, clean up debris, fight off threats. How does lithium deficiency impact these critical guardian cells? Do they still do their job?
This is another really critical piece of the puzzle. Lithium deficiency dramatically alters microglial function, and it's definitely not for the better. It pushes them into what scientists call a reactive pro-inflammatory state. Essentially, they stop being helpful guardians and start contributing to the problem, becoming part of the pathology. And this mirrors precisely what is observed in human Alzheimer's disease brains.
So they turn traitor, almost.
In a way, yes. This snRNA-seq analysis showed a reduction in their normal homeostatic markers. The genes that signal their healthy, supportive state were turned down. Simultaneously, there was a significant increase in genes like apolipoprotein E, which is strongly associated with reactive, disease-associated microglia found in AD. This suggests a fundamental, damaging shift in their very identity and function. These lithium-deficient microglia showed elevated markers of reactivity, things like CD68, GPNMB, LPL, well-known indicators that these cells are activated and promoting inflammation. And when these cells were stimulated, put into a situation where they needed to respond, they released significantly elevated levels of various pro-inflammatory cytokines, things like IL-6, TNF, GCSF, and also immune-activating chemokines like CCL3, CCL4, CCL5, CXCL2.
Chemical alarm bells?
Exactly, like chemical alarm bells and inflammatory messengers just screaming inflammation to the rest of denaturation, amplifying the damage cycle.
So the brain's own cleanup crew essentially goes on strike, or worse, starts making things messier. That sounds like a catastrophic breakdown of order.
It's exactly that, a catastrophic double whammy. Because, crucially, and this is a really critical point for understanding AD progression, microglia isolated from these lithium-deficient mice showed a significantly reduced ability to uptake and degrade amyloid beta 42.
They can't even clean up the plaques.
Exactly. The very process that is essential for clearing those toxic plaques from the brain becomes impaired. So the cells meant to clean up the AD pathology become impaired themselves, losing their critical waste disposal function. Imagine a city's sanitation workers suddenly refusing to pick up trash and then also starting fires in the streets.
That's a nightmare scenario.
It really is. And this creates a detrimental positive feedback loop, a vicious cycle. Lithium deficiency impairs microglial clearance of A. This accelerates amyloid pathology, which then traps more lithium, which further exacerbates inflammation and impairs microglia even more.
Wow.
It's a key mechanism by which lithium deficiency can rapidly accelerate AD progression.
Okay, with such widespread effects, we're talking amyloid, tau, synapses, myelin, microglia. It really makes you wonder, is there a central molecular switch, a single key player that lithium is influencing to cause all this downstream damage? Did the researchers pinpoint such a central regulator?
They absolutely did. And the research points strongly to one specific enzyme, a serine threonine kinase called GSK3, glycogen synthase kinase 3 beta. Now, a kinase, you can think of it as a master enzyme. It adds phosphate groups to other proteins, essentially acting like a molecular switch to turn them on or off, controlling many cellular processes.
Okay.
And lithium deficiency consistently elevated total GSK3 levels and its active form, PTIR216 GSK3. And this wasn't just in one cell type. It was in neurons, oligodendrocytes, and microglia across the different mouse models. This tells us that GSK3 isn't just affected in one place, it's broadly dysregulated across the entire brain when lithium is low. It's like a central command system going haywire. Now, why is GSK3 so important in this context? Because it's a known phosphorylator of tau.
Meaning it adds those phosphates that create the tangles.
Exactly. It directly contributes to the formation of those toxic neurofibrillary tangles, one of the hallmarks of AD. It has also been implicated in app pathology, so it plays a role in the plaques too. And its activity is known to be increased in Alzheimer's disease brains, which makes this finding incredibly relevant. Furthermore, GSK3 plays a crucial role in another fundamental pathway called wnt-catenin signaling. This pathway is important for cell health, growth, differentiation, survival, and it was predicted to be repressed, turned down, by lithium deficiency.
So this one enzyme sits right at the intersection of basically all the major problems.
Pretty much. Amyloid, tau, cell health, inflammation, it's connected to all of it.
So if I'm picturing this right, GSK3 is like this critical overactive master switch. When lithium is deficient, it gets stuck in the on position, unleashing this whole cascade of damaging processes throughout the brain. And if you could somehow flip that switch back,
you'd be well on your way to quieting the storm. And that's exactly what they showed. The importance of GSK3 was confirmed when treatment with specific GSK3 inhibitors drugs designed to block its activity actually reversed many of the pathological consequences of lithium deficiency in the mice.
it worked.
It worked. This included reducing ACT deposition and tau phosphorylation, restoring oligodendrocyte cell numbers and myelin levels, and normalizing that microglial pro-inflammatory activation and even improving their ACT clearance capabilities.
Wow, that's powerful confirmation.
It's a therapeutic revelation, really. It provides incredibly strong evidence that GSK3 is indeed a central mediator of lithium deficiency's devastating effects. It's truly a central player in this entire cascade of decline.
Okay, this research has laid out a clear, alarming problem. Lithium deficiency drives Alzheimer's pathology, likely through this central GSK3 switch. So given how fundamental lithium appears to be, the obvious next step is, can we effectively put it back into the system? But as you mentioned earlier, traditional lithium carbonate, the stuff used in psychiatry for decades, has shown only mixed or even disappointing results in Alzheimer's trials. So what's different here? Why would this approach be any more successful? Why didn't the old stuff work well?
That's a truly critical observation, and it highlights a major scientific hurdle the researchers really elegantly overcame here. They hypothesized that the problem with traditional lithium salts, like lithium carbonate LISP for short, is that they might also bind to amyloid plaques.
Just like the natural lithium does.
Exactly, just like the endogenous lithium. If this happens, it would severely limit their therapeutic effectiveness. It prevents them from reaching where they're actually needed. In the active brain parenchyma, the working tissue, not just getting stuck in those inert, damaging plaques, it's like having a medicine that gets absorbed by a sponge before it can even reach the actual wound it's supposed to treat. So their scientific approach to identifying the right lithium, better form, was ingenious. They systematically measured the electrical conductivity of 16 different lithium salts. Their reasoning was quite clever. Salts with lower ionization, meaning they form fewer charged particles in solution, would have reduced electrostatic interaction. They'd be less sticky.
Less likely to bind to the charged amyloid.
Precisely, and thus less likely to bind to amyloid. What they found was truly groundbreaking. Organic lithium salts, and particularly one called lithium orotate, or LEO, showed the lowest conductance across various concentrations. This indicated significantly reduced ionization compared to inorganic salts like lithium carbonate.
So lithium orotate is potentially a smarter form of lithium, less likely to get caught in that amyloid trap, allowing it to actually get to where it's needed in the brain tissue itself.
Exactly. It's all about targeted delivery, getting past the roadblock. To confirm this, they performed in vitro binding assays. They use synthetic human Ev142 fibrils and oligomers, essentially, testing how well the different lithium forms stuck to lab-made amyloid in a test tube. And indeed, lithium orotate exhibited significantly lower binding affinity, it stuck less, than lithium carbonate. This was true across both physiological and pharmacological concentration ranges. This was a direct, irrefutable demonstration of its plaque-evading property. And then they confirmed this in vivo in live animals, which is the crucial next step. They administered low-dose LiO or LIGHTC to 80 mouse models, and they observed that LIC, the carbonate form, was highly concentrated in the AT plaques, just as they suspected it would be, while LiO, the orotate form, was concentrated to a much smaller extent in the plaques.
So it wasn't getting trapped as much.
Right. And more importantly, LiO, but not ALA, significantly elevated the lithium levels in the parenchyma in the non-plaque fractions of the brain. This directly proved its plaque-evading capability. It could actually get to the parts of the brain where it was needed to exert its therapeutic effects.
That is a critical distinction.
It really is. It's not just about having lithium available systemically. It's about having the right form of lithium, one that can actually reach its targets within the brain without being trapped and rendered ineffective by the amyloid pathology itself. It's the difference between, say, an unguided missile and a precision strike.
That's a huge breakthrough in itself, just finding a delivery method that bypasses the problem. But the ultimate question, of course, is does this smarter lithium, this lithium orotate, does it actually work? Does it combat the AD pathology and those devastating consequences we discussed earlier in living systems?
The results here are remarkably compelling, and they truly offer a profound glimmer of hope, I think. When lithium orotate was administered at a low physiological dose to adult AD mouse models, it almost completely prevented both a plaque deposition and phosphatase accumulation.
Completely prevented.
Almost completely prevented. Think about that. Near total prevention of the two defining protein pathologies of Alzheimer's. In stark contrast, lithium carbonate at the same low dose, or a sodium orotate control used to make sure the effect was from lithium, not the orotate part, neither of those had any significant effect. This really points to lithium orotate's specific and potent efficacy at these low doses. Now, even more exciting perhaps is that Leo demonstrated the ability to reverse more advanced established pathology.
Right, reverse, not just present.
Yes, reverse. They administered Leo to older AD mice that already had widespread apocket deposition. Their brains were already heavily burdened by the disease. And it significantly reduced the A-plaque burden and the density of phosphatase-positive neurofibrillary tangle-like structures in the hippocampus by about 70%.
70% reduction of existing damage. That's incredible.
A massive reduction in existing pathology, not just preventing new formation. And again, IIC, the carbonate form, remained ineffective at these doses.
That's absolutely stunning. Not just preventing the damage from starting, but actively reversing damage that's already there. That really shifts the whole conversation from just prevention to potential treatment, doesn't it?
It really does. And the impact was holistic. It addressed multiple facets of the AD pathology. Beyond just the protein aggregates, LIO also increased the expression of that postsynaptic protein, PSD95.
Crucial for synapses.
Right, crucial for the structure and function of synapses. It boosted myelin-associated MBP immunoreactivity, critical for healthy nerve insulation. It elevated oligodendrocyte cell numbers, the cells that produce that myelin. And furthermore, it effectively suppressed microgliosis and astrogliosis, those indicators of neuroinflammation in the aged AD mice. It essentially quieted the brain's internal alarm system that had gone haywire. And consistent with our earlier discussion about GSK3 being that central regulator, low-dose LIO, but not LIO, reduced total and activated GSK3 levels. And it elevated nuclear catenin, further supporting its mechanism of action through inhibiting this key kinase.
It hit the master switch.
It appears to hit the master switch. This demonstrates LIO's potent ability to not only prevent, but also to significantly reverse core AD pathologies. It acts on multiple fronts simultaneously to restore brain health. It's a remarkably comprehensive and elegant approach from a single element when delivered correctly.
Ultimately, though, the goal in fighting Alzheimer's is to preserve and restore cognitive function, to protect those precious memories, that ability to connect with loved ones. Does lithium orotate deliver on that front? Does it actually restore clarity of thought in these models?
Absolutely. And the impact on cognitive function was truly one of the most exciting and hopeful outcomes of this entire study. Lithium orotate, even at its lowest physiological dose, almost completely reversed memory loss in the AD mouse models compared to the untreated controls.
Reversed memory loss.
Almost completely reversed it. This wasn't subjective, it was measured through various standard behavioral tests showing remarkable recovery of function. It also significantly improved learning and spatial memory in aging J20 mice that already had advanced amyloid pathology. This means it wasn't just preventing decline in younger mice, it was actively improving function in brains already compromised by established disease.
That offers real hope for people already experiencing symptoms.
It really does. Imagine the potential for people with early-stage cognitive impairment or even mild dementia. What's more, and this is another crucial point for all of us, I think Leo wasn't only beneficial for the AD models. It prevented age-related loss of dendritic spines in the hippocampal neurons, those crucial connection points in normal aging mice. And it largely reversed the decline in learning and memory associated with normal aging in wild-type mice.
So,
It could potentially help maintain cognitive sharpness and slow down typical age-related memory decline in general, not just in the context of Alzheimer's itself. That's a huge implication for all of us as we get older.
It certainly is. And importantly, these cognitive improvements were not due to changes in basic motor function or sensory perception. The mice weren't just moving faster or seeing better. The effects were directly on memory and learning pathways on the very core of cognition.
That's very clear then.
Yeah. This is incredibly promising. It suggests that targeted lithium supplementation with the right formulation like orotate could potentially restore cognitive abilities that are typically lost to aging and Alzheimer's. It offers a real glimmer of hope for maintaining mental clarity, preserving those precious memories well into later life. It's a powerful statement about the potential of the therapy to protect what makes us, us.
Now, with any therapeutic approach, especially one you might consider for long-term conditions like brain health, safety is absolutely paramount. Has to be. Are there concerns with lithium orotate, particularly given that traditional lithium carbonate can have some significant side effects, especially with long-term use? What did the research show regarding its safety profile?
This study meticulously addressed that critical question. It's absolutely vital for any potential future human applications, so they were extremely thorough. Long-term administration of low-dose lithium orotate to both aging wild-type mice and AD mouse models did not alter serum levels of key markers for kidney and thyroid function. Things like blood urea, nitrogen, creatinine, or thyroid-stimulating hormone, TSH.
And those are the common concerns with traditional lithium, right? Kidney and thyroid.
Kidney and thyroid toxicity are common and serious limitations of the pharmacological doses of traditional lithium carbonate. They've historically limited its broader long-term use for conditions like Alzheimer's or even bipolar disorder in some patients. So the absence of these adverse effects in the LiO-treated mice at these effective doses is a major, major positive indicator for its potential safety. Another key point here is the dose level. The doses of LiO used maintain serum and cortical lithium levels in the endogenous range.
Meaning the natural range.
Yes, the natural, typical range found in the body. This means the mice weren't given excessively high, unphysiological amounts. They were essentially restoring lithium to its natural optimal levels, rather than pushing it into a pharmacological overdose, which is often the approach with traditional lithium therapy. This subtle but crucial difference in dosing and formulation seems to make all the difference for both safety and efficacy, and connecting these animal findings back to human health again. The researchers looked at their human data. In normal aging individuals without MCI or AD, higher natural cortical lithium levels, more lithium naturally present in their brain tissue, correlated positively with cognitive test scores.
Really?
Yes. Specifically for working memory and performance on the Mini Mental State Examination, the MMSE, a common cognitive screening tool. This strengthens the idea that simply maintaining healthy, optimal brain lithium levels is important for cognitive resilience in humans, even in the absence of overt disease.
It supports the whole premise.
Exactly. These findings collectively open a very exciting avenue for the prevention and treatment of AD. It suggests a potentially natural and well-tolerated approach that could have broad implications for senior wellness and maintaining sharp cognitive health as we all age. ### Archer Afro.
So wrapping this up, what does this all mean for us here and now? This deep dive into this groundbreaking Harvard study published in Nature, it reveals a truly compelling and frankly alarming story. Lithium deficiency in the brain appears to be not just a symptom, but an early and significant factor in the pathogenesis, the development of Alzheimer's disease. It contributes to this devastating cascade of issues from amyloid plaques and tau tangles to widespread neural inflammation and ultimately profound cognitive decline.
And critically, this research points to more than just identifying the problem. It really offers a genuinely practical and rather elegant solution. The study highlights that a specific form, lithium orotate, seems able to bypass that amyloid sequestration, that insidious, sticky trap we discussed. It effectively replenishes brain lithium right where it's needed most. And this targeted approach was shown, at least in these comprehensive mouse models, to not only prevent but also to significantly reverse established AD pathology. It quelled destructive neuroinflammation and most remarkably appeared to restore memory and cognitive function even in advanced stages. And all this without evidence of the typical lithium toxicity at these low physiological doses. It suggests a precise, intelligent approach to a profoundly complex and devastating problem.
This research really does suggest a potential paradigm shift, doesn't it? In how we think about brain health, aging, maybe even the future of Alzheimer's prevention and treatment. Could maintaining optimal physiological levels of an element like lithium, maybe through careful dietary considerations
or perhaps targeted low-dose supplementation with something like lithium orotate. Could that become a proactive and accessible strategy for preventing, or maybe even treating, Alzheimer's, for preserving sharp, vibrant cognitive function well into our later years? It's a question that could genuinely change so many lives if the answer is yes.
And this isn't just a theoretical question for the distant future, I think. It raises an important consideration for all of us right now. Given that lithium is naturally present in our environment, it's often found in varying trace amounts in drinking water all around the globe. Could subtle, chronic deficiencies in our modern diets or perhaps our treated water sources be a contributing, yet largely unrecognized, factor in the global rise of dementia?
That's a big question.
It is. There have been fascinating epidemiological studies over the years hinting at correlations between natural lithium levels and local water supplies and community mental health statistics, even things like suicide rates or dementia prevalence. But this research, perhaps for the first time, offers a direct biological mechanism for why those trace amounts could be so incredibly vital for brain health. It really urges us to scrutinize what might be missing from our environmental puzzle, especially in an age where, our diets are increasingly processed and our water sources are often heavily treated, sometimes stripping natural minerals. Could this tiny overlooked element be a critical yet missing piece in the much larger health crisis of global dementia? It truly invites us to consider how the smallest things in our world, the trace elements we barely think about, can have the biggest, most profound consequences for our long-term cognitive well-being.